Hello everyone, welcome back to another video from Shomu's Biology. In this particular video lecture, we are going to talk about actin filaments. What are actin filaments? How they look like? What is the structure of actin filament? What are the components of actin filament? And also, you will see the important properties of actin filament, that is nucleation as well as polymerization, depolymerization and everything in between, which is known as actin polymer or actin filament uh, treadmilling. So, we will see all these things in this today's lecture. So, let us start talking about the actin filament now. I will start with this discussion of actin filament. First of all, this actin filament is a part of cytoskeleton. Okay. If you recall our understanding of cytoskeleton, we have talked about cytoskeleton earlier in the cell organelle section. I told you that cytoskeleton are the structural component to the cell, generally made with proteins, which provides enormous structural strength to a cell. Either they provide the cell of a basic cellular shape and structure to be maintained, and also they have other properties like help in the motility and also organizing vesicles and also the movement of organelles inside the cell. All these things are done by the cytoskeleton. Now, among cytoskeleton elements, we have different types. We have different kinds of cytoskeleton element in the stator section. That means the element which generally is providing structural integrity, which is stagnant to a place, not movable. That kind of thing is actin filament. Now, actin filament is one of the most predominantly found cytoskeleton element in the cell, and in this cytoskeleton element, what I say is that they are made with proteins. So, the very first thing that I want you to know, they are made with proteins. What kind of proteins are they made with? Obviously, in this case, actin is the name of the protein they are made with. Actin, A-C-T-I-N. Now, this actin protein is just like a simple small round shaped protein, and this single round shape is known as the actin monomer. You need to keep it in your mind, the actin monomer. Now, this actin protein can function without, it cannot function without ATP. So, the actin monomer can either associate it to ATP or it can associate to ADP. Both of these are possible. If the actin is associated to ATP, that is known as a part of active monomer of actin, and if it is associated to ADP, then it is inactive. It is inactive part of the actin filament. So, when the actin monomer is associated to ATP, that is known as the active part of the actin. When it is associated to ADP, known as inactive part of the actin. Now, the question is, I told you that the actin filaments are like one of the predominantly found structural integrity components or structural component of a cytoskeletal element among the cell. So, what happens is that if you consider that this is the cell, this is how the cell look like. Inside the cell, we have these actin filament structures forming, and these actin filament structures, they continue to form and interact towards the cell membrane, and it can push the cell membrane to move towards any particular direction. And with this movement of a cell membrane with the cytosol in a particular direction, that can cause the formation of flagella, not pseudopod structures, not flagella, formation of pseudopod structures, cytosolic extension that helps the cell, particularly the single cell organisms like for example amoeba, let's say, to move towards their food resources. This is one such example, practical examples of why actin filament is so important. But what we want to understand is the structure of this actin filament. Now, as I told you that this actin as a monomer is non-functional, is without any use, but actin as a polymer, as a fiber, give rise to a lot of important features, including the structural strength to the cell, including the movement of cytoplasm or membrane towards a particular direction, just in case of amoeba example. Okay, so what we can clearly see here, what we are going to talk about here is how exactly this monomer of actin, so this monomer of actin turn to the actin polymer, how the monomer is converted to actin polymer. This is very important. For this process, actually they need one step known as nucleation. So, if you are reading this for BSc or MSc standard as well, you note that actin nucleation is another very important concept that you should understand. Now what is actin nucleation? Let's imagine that in an environment we have actin monomers floating around in the cell. The monomers are like glue and they should have a glue to adhere to each other. Now usually the monomers are without any sort of glue, right? And we say the actin without the polymerizing protein, okay? R means meaning the actin without the glue. So without polymerization protein, the actin monomers although are present in the environment, but they will not stick to each other, they will not convert into a polymer. They will only convert into a polymer when they have that molecular glue structure to them, okay. ATP once associated to actin provides that glue to that actin. So I consider this as a molecular glue like that. So the actin here, so remember here at this moment we have actin monomer but with ATP. Such actin monomers with ATP together and more than 3 to 5 such monomers once stitched together, then that is, so let's say 5 such monomers stitched together due to the presence of actin associated to them is known as the nucleation of actin. The nucleation of actin. So this is the nucleation of actin, but the nucleation of actin is again not possible if there is no nucleation protein, and the nucleation protein in case of actin is known as ARP2/3 complex. Actin nucleation protein complexes are needed like ARP2, ARP3. This ARP2/3 complex is known as nucleation proteins, and they along with the ATP actin more than 3 to 5 and sometimes also 8 such actin monomers with ATP units has a molecular glue structure with the presence of ARP2 and 3 start to interact to each other in different orientation. And once 5 such, at least minimum of that 3 to 5, that is the threshold. So that amount of ATP associated actin stitched together with the presence of ARP2/3 complex, the nucleation is done. And once the nucleation is done, then the process of actin polymerization will begin. Without nucleation, actin polymerization cannot begin. Okay, so once the nucleation will be done based on the presence of nucleation proteins and the ATP actin, the threshold is passed, and then the nucleation is complete. And once the nucleation is complete, then what would happen at the end of the nucleation? We have some of these actin monomers stitched like this. Okay, they have ATP associated to them. And they also have this ARP2/3 complex, and this is the nucleated form of actin. This is a first functional form of actin that is ready. So nucleation is done, this form of actin is ready. But now just the nucleation is passed, and this is known as a critical concentration of actin monomers. F actin monomers means actin along with ATP, that monomer, the number of that monomer is known as the critical concentration. Let's say the critical concentration for that is 5. So once we have 5 ATP actin along with the presence of ARP2/3 complex, they are going to be marked as the completion of the threshold value and the nucleation process is complete. Then only the polymerization would begin. Now the next step is polymerization. Remember, next step is polymerization. We'll start the polymerization. Now after that, the polymerization process is pretty straightforward, and this is where we come to the second point of trade milling. We come to the point of trade milling. Okay, so in this point of trade milling, what we exactly mean by trade milling here is that these actins that are present there. Let me draw this actin. So the actin, they have two different ends. We have this end. Okay, this is one end. Okay, the barbed end basically. This is where the actin polymerization is full swing, and there's another one is the pointed end. But what I mean to say is that try to understand this concept. So this is the black colored ones are the actin with ATP. They are the actin with ATP. And let's say we have the green colored ones. These are the actin with ADP. Now, as I told you earlier, the actin can stay with either ATP in the active form or ADP in the inactive form. So while this polymer is built, there is always two terminal. In one terminal, ATP associated actins are present predominantly. In the other terminal, ADP associated actins are present predominantly. The site where ATP associated actins are present are known as the plus end, because this is the growing end. This is the growing end. While the other end, the opposite end, where the ADP associated actins are present, known as the minus end or shrinking end. Okay. So we have a growing end, we have a shrinking end. We have two separate ends like this. And in a polymer structure of actin, the new actin monomers continue to be added to the plus end. Okay. At ATP associated forms are continuously added to the plus end. So the new actins are always being added here. The ATP associated actins are always being added here. But while we continue to grow, from the opposite end, the ATP is hydrolyzed to ADP. Now the ADP associated actins start dissociating. So dissociation start to take place from the minus end, association start to take place from the plus end. In this way, the actin goes to the plus end, shrinks from the minus end, goes to the plus end, shrinks from the minus end. So this is a dynamics of actin, and this is known as the treadmilling effect. So in treadmill, what happens? You run and the path ends, you shrinks down to the earlier position. So this is treadmilling, this is shift, again moving towards plus end, again minus end. So this way the actin polymer grows and shrinks, growing from the plus end, shrinking from the minus end. This is actin treadmilling, and actively this treadmilling effect continue to form, continue to grow. Okay, this is a part of the actin polymerization. In that way, now you understand how exactly this actin filament can push the membrane towards a particular direction, because obviously the plus end is towards the membrane. So it's pushing the membrane along with the cytosol in one particular direction, while the cell try to transfer the, start to form the pseudopods to capture food or prey. This is possible. Okay, this is the simple idea. But this polymerization never ends here, because this polymerization, particularly for the plus end, to stabilize the plus end, we need a capping protein. Okay, and actually this capping is possible for both stabilization as well as destabilization in case of this sort of actin filament polymerization. So for the positive format of capping, that is a capping protein involved in here, and also in some other hand, I'm going to talk about the dissociation of actin filaments. There is also another capping method available there in the dissociation of actin filament as well. So this capping point of view is important because it is supposed to protect the plus end, and that is the name. Here the name is cap Z. Cap Z or cap Z is the protein that adds a cap to the growing end or the plus end, you can say. Okay, and that helps to stabilize the structure in the plus end. Similarly, there is a capping event in the minus end to destabilize the structure, and that is by gelsolin. Gelsolin is another protein that is responsible for destabilization near the capping of the minus end of the growing and shrinking microtubule in the dynamic growing and shrinking actin filament, the dynamic actin filament. Okay, so cap Z adds a cap to stabilize the plus end, and gelsolin's effect in the negative terminal to destabilize. So these are the two set of proteins involved as a cap here. Now remember one more thing, because we are looking at a very dynamic nature, this is not fixed. The length of actin filament is not fixed because it's growing and shrinking. You cannot give an idea about the length of the actin filament. You can tell about the diameter of the actin monomer, this polymer's diameter, that you can tell, but not about the length of the actin, because depending upon cell's requirement, it can vary itself, it can change itself. Okay, that's giving the dynamicity to the cell to modify its structure as well. One more thing that you need to understand is that between this actin, there is actin monomers association to, remember, actin monomers association to ATP, and then actin association to ADP. We know once associated to ATP, that is the active form, associated to ADP, inactive form. But who helps to substitute the ATP with ADP, right? Because one moment the actin is associated to ATP, that is the molecular glue, the work is done, ATP is hydrolyzed to ADP. Now the ADP should be released new ATP should be associated. So a requirement of ATP ADP substitution is needed. Who does that? Another important protein does that, and the name of that protein is profilin. So the name of that protein is profilin, and what profilin does is the substitution of ATP and ADP in an actin. So the moment ATP is hydrolyzed to ADP, the ADP is substituted with a fresh copy of ATP to the actin done by profilin. So profilin actually helps in the process of polymerization of actin. Actin polymerization is helped by the profilin. And one more thing, as profilin can act on single actin monomers, so sequestration is also possible by the profilin. Remember that. Okay, sequestration is also possible. Another protein is involved there who can do separation of monomers from each other, that is known as thymosin. So remember these two proteins named profilin and thymosin. Both of them involved in the process of sequestration of individual monomers of actin from the actin filament. So largely the profilin helps in the process of polymerization. So as there is a protein help in the process of polymerization, remember one more thing, that once this process is done, we know that the actin associated to ATP and then it is hydrolyzed to ADP, and what we simply do, we do this exchange of ADP association of new ATP done by profilin. Okay. Now what else we need to know? We need to know one important point here is regarding the polymerization. We have done the polymerization, right? So this is our actin polymer that is formed. Now the question is single length, because the actin polymer is the thinnest among all the three types of cytoskeleton element that is found in the cell. One is the microfilament, means the actin filament, the same thing. Microfilament and actin filaments are same thing, the thinnest. Then we have intermediate diameter, intermediate filaments. Then we have microtubules, the largest diameter. So is it possible to bundle this microtubule? Is it possible to bundle this actin filament or microfilament? Sorry, I use microtubule more often. This is a microfilament. So is it possible to bundle them? Do you have any protein to bundle them and hold them structures together, like two strands, two polymers of actin, and bundle them? Is it possible? This is one thing regarding bundling. Bundling possible or not? And second thing, is it possible to have a branching like this, branching of this, branching, okay? Is it possible? These are two questions that always come in our mind. Is bundling possible with microfilaments or actin filaments, or is branching possible with actin filaments? The answer to both of this are yes, this is possible. This bundling process is done by a protein known as fimbrine. And this branching process is done by alpha actinin. So there are name of a lot of proteins in this particular section, I understand. It might take some time for you to grasp them, but you can write it down in your notebook too, because these are some information you will never get in any other video here. Okay. So the bundling event is possible to make the structure more strong, and that's why we have fimbrine protein involved. But for branching, segmentation, that is also possible by the protein alpha actinin. Okay, both of them involved in their own approaches. Now apart from this, this is all about polymerization, and remember we know that this fact that the polymerization is from the plus end, obviously. So from the minus end there will be depolymerization. So do we have any depolymerization promoter? Yes, we do have a depolymerization promoter, and that is known as ADF cofilin. Cofilin protein is cofilin don't like microfilaments, they don't like actin filament, they always try to destabilize it. So ADF cofilin is the protein complex that involved in the process of actin depolymerization or actin filament depolymerization. It always wants the actin to shed off from the minus end. That is the important feature of cofilin. So if you look at cophilin and profilin, the roles are different. Profilin helps in the polymerization of actin, while cophilin helps in depolymerization of actin. Although we know that along with I've written here the destabilized sequestration, along with thymosin, the profilin can also sequester individual monomers of actin, which is very important, that after the polymerization is done, depolymerization is done, treadmilling event is over. So the individual actin monomers need to be sequestered, need to be stored, so that we can utilize them to make polymer again. That is also done by the thymosin and profilin, but majorly the role of profilin is the polymerization, while the role of cophilin is the job of destabilization in actin filaments. So that is in a sense overall idea about the actin filament. And regarding the function, if I want to talk about it, I already told you that providing mechanical strength to the cell, and also that this actin filaments are connected to the different cytoskeleton elements in the cell, okay, and also help in the cytosolic streaming. It is involved in the process of the extension of the cell cytosol, formation of pseudopods. Okay, so these are all the important features that we know about the actin. And one more feature is this muscle contraction. The actin and myosin is the motor element and actin is the stator element. Both stay together. The actin filaments build the path on top of which the myosin try to slide, and as a result of which muscle contraction is possible, particularly in the skeletal muscle. The skeletal muscle contraction is possible due to the actin myosin slide, okay, with the power stroke action model, which we will discuss in a separate video later on. But these are the features and functions of actin filaments in the cell. I believe this video gives you a clear idea and a lot of information regarding the actin filaments. If you like this video, please hit the like button, share this video with your friends, subscribe to this channel to get more videos like that in future. Thank you. Bye.